Okay, and now we're on to our second lecture in simple linear models, which is about interactions. So again, no readings for class, but I'm going to point out the two books that I mentioned before that are really great, and then also a paper that we're going to talk about in this lecture that you may want to look at for more information. So we're going to talk about factorial experiments, which I think are a super powerful way for trying to understand how your independent variables, your x's, either add or interact to affect your y's or your independent variables. So I just really love these experiments a lot. They're really powerful. And the general idea of how it works is that you have, say, two treatments. Those are your two factors. And you would have treatment one, you'd either have it or you wouldn't. So plus or minus treatment one. And treatment two would either be plus or minus treatment two. And then you would have your resulting experimental units, which could be plots or could be, anything, where you have some with both treatment factors. So you have plus treatment two plus treatment one. Two units where you have one of the treatments and not the other. So plus treatment one minus treatment two and vice versa. And then you have your final one where you have neither treatment two or treatment one, and that's considered your control. And so we're going to talk about factorial ANOVAs. These are the simplest form of all of these interactions, but they can get much more complicated, of course. But we're going to just start here. And so you're going to factorial ANOVAs have dependent variables y, one dependent variable y that's continuous, and an independent variable x that's categorical and interacting. And so the interactions here measure the joint effect of the main effects of A and B. So in the previous case, that would be treatment one and treatment two. And it identifies if response of A is dependent on your level of B. So here's your equation. Again, don't worry about it too much because these things start to get pretty complicated. But just know that one really important thing to know about this is that if you have a significant interaction, then your main effects are not interpretable without clarification. So if you have a significant interaction, you need to look at your data, look at what's going on, and figure out what your result is without just looking at your main effects. And these interactions are actually really common in biology, and so they're really important to understand how to interpret them. And you can add more factors. It doesn't just have to be two, right? You can do treatment three, for example, as well, and look at the interaction of all three. But this starts to require a larger sample size, so larger N. So just keep that in mind. And so what do interactions look like? I apologize, this figure is a little blurry, but it really shows what I'm trying to show with these interactions is that, okay, so you have factor A and factor B. Those are your two treatments. And you have levels 1 and 2, so high and low for both, or on and off, right? Plus or minus. And so interactions you would see either in the first panel, you could see that the results completely cross. So that's one type of interaction. You can also have it where for the slopes, one has no change in slope. So your red line here is flat and your blue line is diverging from that line. And so that would be a type of interaction. That's what you might see a significant interaction to look like. Or finally, you could see that they both there is a slope between both your pink and your blue lines here, but they're diverging. And so if you get a significant interaction, this is what they're going to look like, one of these. If the two lines were parallel all the way, then there would be no interaction. And so how do factors add? I apologize, it's difficult to see the bottom of that, but this is from the Harbold 2011 Ecology Letters paper. And so, right, we were talking about how there would be, if there's an interaction or not. So you can see this, I'll just orient you. So here at the bottom, right here, this is your response variable is on your y, of course, and then here you have no treatment addition. So this is your control. This is when you add one. This is when you add two, the other one independently, and this is when you add both together. And so there's lots of ways, lots of results you could get from that. So if you see this dotted line for 1 and your second line looks like this additive response, then that means that there's no interaction. However, if you see this line and this line, that's less than this additive response, and that's going to be an interaction, but it's a sub-additive interaction because those lines are converging. And then in the second case is if you see your first line looking like this and your second line is above the slope of the additive line, then that means your interaction is super additive or multiplicative. And so basically here it would say that the whole is more than the sum of its parts, right? If you add treatment 1 and treatment 2, you get whatever your result is, but when you add them both together, you get more than what happens if you add the single ones together. Subadditive is the opposite. You add a single one and a single one. If you add them both, you get less than what you might expect if you added them at the same time. So I think it's really interesting to think about how factors add and what interactions look like. So again, parallel lines mean no interactions, and then your diverging or converging results mean an interaction, but what do those interactions mean? Are they additive or super additive? And so you can look at this paper, Harpel 2011, et al. 2011, and see all sorts of different types of these, but this is the simplest and most straightforward types of interactions. So today we're going to be looking at data on biomass of different plants under either a nitrogen addition, a phosphorus addition, or both, or neither. So a factorial experiment of N and P. And this is the best picture I could find for biomass. So it's going to be pretty data. And so here's what our experimental design looks like. So biomass differences, we're going to be looking at the biomass differences for plant under either N and P together, N alone, P alone, or neither. And so this is just a little depiction of what this experiment actually looks like. And so here's the data. And I wanted to show you this purposely because you can see that the way that you run these models, these factorial models, is that you have to create these indicator columns. So you have your N and P columns, right? Because it's either N, yes or no, or P, yes or no. And that those columns mirror the experimental design on the previous slide. So zeros indicate that the treatment is not applied in that plot or unit, and ones indicate that that treatment is applied. So your control plots are going to be 0, 0, as you can see here. And then nitrogen plots are going to be 1, 0, phosphorus plots are going to be 0, 1, and nitrogen and phosphorus together are going to be 1, 1. And so that's how you need to code. These indicator variables are how you code these models for your analysis in R. And so let's look at this data again. How does the addition of nitrogen and or phosphorus influence plant biomass? So you could look at just all of the treatments here on the bottom. If you didn't, you could look at all the treatments, your control, NP and NP. And so you could run a simple linear model on this data, right? You don't necessarily have to run a factorial experiment, but it's not going to give you the same results. So say you just run a simple linear model and you want to look at biomass as a function of treatment, because remember we had a treatment column that was actually the labels of the treatment. And here you can see that nitrogen is significant, and so is nitrogen and phosphorus together. So that's one way you could look at this data, but this isn't a factorial. This doesn't include an interaction. Okay, but you could also do a factorial ANOVA instead of just a regular linear model or a regular ANOVA, and to look at does the interaction of nitrogen and phosphorus influence the biomass of plants. And so to do this, you have to go back to your N and P variables, your indicator variables, and you code those in as your X variables. And so then when you get this, you see the same, you see a similar result, but here the NP treatment isn't the NP plot, it's the interaction of N and P. And so again, remember, you can't interpret that nitrogen has a significant effect because you have an interaction of nitrogen and phosphorus. So let me show you what I mean by that. So when you look at this here, you have your nitrogen addition on the x-axis, and you have your biomass on the y. And now you have your phosphorus, your second factor, as the colors. And so it doesn't make sense to say that nitrogen addition alone has an effect because it's in relation to phosphorus. So when you have no nitrogen and no phosphorus, that's not different than those bars overlap as to when you have no nitrogen and phosphorus. And so, but you can see these lines diverge for when you have nitrogen additions, and then if you have phosphorus in the blue, then you get a much higher response than if you have no phosphorus. So you can see you really need to interpret this graph with the interaction in mind. Just saying nitrogen addition wouldn't really make any sense because that would be like lumping that nitrogen addition 1 column, but you can see it really depends on if you have phosphorus or not. Okay, so I want to talk about higher order interactions briefly. So when you write n times p with those indicator variables, that's the same thing as saying n plus p plus n colon p. That n colon p is like writing that highest interaction. And so the way it works is that, again, under the hood, the variance is separated. So you look at the effect of n and p together as well as the effect of n and p alone. And so that's how these models work. So that's why you can separate it as n plus p plus n colon p. And this is actually really important because when you start to get higher order interactions, for example, more than 2 factors, you can start to eliminate your highest interactions if they're not significant by reverse elimination. So what I mean by that is if your highest order interaction, for example, in our case, n times p, but in another case, so that would be the n colon p would be your highest order interaction. If you had 3, it would be n, and your factors were n, p, and k. Your highest order interaction would be n colon p colon k. If that highest interaction isn't significant, you can't interpret the rest of it because that higher order interaction is sucking up all of your variance. And so what you need to do is get rid of that highest interaction and then work your way back. So for example, you would start with n times p times k, and then say that 3-way interaction isn't significant. You would go to the next model that's then n times p times k minus that 3-way interaction, which is n colon p colon k. So that would be all interactions. That would be all interactions including n, p, and k separately, n times p times k, but not that 3-way interaction. And you sort of work your way back from there. And so with that, please complete the quiz on Canvas, and that's the last lecture for this week.